The giant tube worm, Riftia pachyptila, lives in symbiosis with the chemoautotrophic gamma protobacterium cand. In Riftia Persephone, symbionts are released back into the environment upon the host's death in high-pressure experiments. While microbial fouling is not involved in trophosome degradation, therefore we examine the antimicrobial effect on the tube worm's trophosome and skin. The growth of all 4 tested gram-positive with only 1 tested gram-negative bacterial strains was inhibited by freshly fixed degrading trophosome, incubated up to 10 days in either warm or cold temperatures. While no effect on the Saccharomyces cerevisiae were observed, the skin did show antimicrobial effects. The liquid chromatography mass spectrometric analysis of the ethanol supernatant of the fixed trophosomes led to tentative identification of phospholipids. Number 1, palmitoyl, 2, lysophosphatidylmethylamine, 2, palmitoyl, 1, lysophospholipid linolelamine, and the free fatty acids palmitoleic, palmitic, oleic acid, which are all known for having antimicrobial effects. As a result of the tissue and autoanalysis, the abundance of the free fatty acids increased with longer incubation time of the trophosome samples. This correlated with an increasing growth of the inhibition of Bacillus subtilis and Listeria welchemeri, but not of the other bacterial strains. Therefore, the free fatty acids produced upon the host degradation could be the cause of the inhibition of at least 2 bacterial strains. Since the discovery of the giant tube worm in the deep sea hydrothermal vents of the Galapagos Rift in 1977, the mutualism between the sessile tumor Riftia pachyptila vestibutifera sibolanidae, short for Riftia, and the sulfur-oxidizing gamma protobacterial symbionts cand, Riftia Persephone, or short for Riftia, has been one of the most extensively studied deep-sea symbioses. The meganome of genome of Riftia encodes genes for sulfur oxidation and carbon fixation, but also genes for tricarboxylic acid, TCA, cycle, fructose degradation, glycolysis, as well as phosphotransferase system and the ABC transporters. The latter are indicative of heterotrophic lifestyle that is assumed to play a role outside the host's absence of the sulfide. Also, genes for chemotactic abilities include flagellar proteins, chemotaxis regulators, and motility accessory factors required to the survival outside the host were detected in the megagenome. Riftia is located in the trunk of the adult host's body in a multilobal organ, the trophosome enclosed in its host cells called bacteriocytes. Host bacteriocytes and symbionts exhibit coordinated cell cycle with terminal differentiation. Dividing rods of unpotent bacteriocytes acting as stem cells are located in the central zone of either each lobule. A small cocci and large cocci are located in the semi-differentiated bacteriocytes in the median zone, while degrading large cocci in the terminal bacteriocytes of the peripheral zone either enter apoptosis after digesting the symbiont, nourishment of the gutless host by the symbiont through the release of the fixed organ carbon. Symbiont acquisition is the horizontal in each generation anew. Symbionts invade the settle larvae, and small juveniles are shown by fluorescence in situ hybridization, or FISH, using 3 specifically designated symbiotic-specific oligonucleotide probes. Environmental symbiotics are detected with 16S RNA-specific PCR and FISH on artificial devices deployed in tuber clumps next to the clumps as far away from the clumps on the basalt as well as filter seawater from the pelagial. Recently, we could show the experimental high-pressure vessel in the Riftia actively escapes the dead trophosome tissue and it recruits surface upon which it proliferates. The escape time was determined in a series of incubations simulating either vent cessation with cold ambient deep sea conditions to half a day or 6 days warm hydrothermal vent conditions with sulfide flow through the system of half a day. The disintegration of the symbiont's membranes was studied in the transmission of electron microscopy, or TEM. The experiments reveal that under warm vent conditions, most of the symbiont membranes were ruptured by the symbionts, therefore unambiguously dead after 1 day. The symbiont's decay was decelerated under the cold deep condition, the membranes still intact in the days. Numerous studies have shown that no other microbes colonized trophozoans in living animals apart from Riftia. Surprisingly, preliminary FISH using the symbiotic-specific and bacteria probe mix EUB338, 1, 2, and 3, which targets the most bacteria simultaneously on the incubated trophozone pieces, revealed no microbial fouling during the true degradation in or escape experiments. Therefore, we investigated whether the gram-positive and gram-negative bacterial strains of fungus were inhibited by the growth due to the presence of the trophosome pieces of the ethanol supernatants derived from the fixation. The trophosome samples were either freshly collected, representing metabolism in most host symbionts, or incubated under a simulated deep sea hydrothermal vent conditions, representing dead host and living symbionts initially, or dead host symbionts in later time points. Further, we analyzed the chemical composition of the ethanol supernatants of fixed, fresh, incubated trophosome samples with liquid chromatography mass spectrometer, or LCMS, and tested whether the abundance of the identified compounds correlates with the inhibiting effects of the growth of the tested microbes.